Lesson 18, we will be able to describe the process and stages of mitosis. My goal for today is going to be this. This lesson, I'm going to quickly go through mitosis, and then I'm going to give you guys some questions to do on style. Then at 2 o'clock, about 2 o'clock-ish, 2:20, when you guys would have period 6, what we'll do is we'll go through meiosis. And we'll quickly talk about what the difference between the first one and the second one is, and then the rest. And so this is going to be a pretty short lesson because it's not too much to go through. Let's actually go backwards a sec. I want to go all the way back to this. We've actually talked about mitosis and meiosis before. We said that mitosis is the process, most body cells split using mitosis. And the idea of mitosis is that each cell gets the same copy of chromosomes. They both start with 46, ends with 46, ends with 46. The cells that do mitosis, they're called somatic cells. We said that meiosis is different. Meiosis is instead the daughter cells, or the afterward cells, actually end up with less chromosomes. You go from 46 down to 23. That's half as many. We call these cells gametes. And then we talked briefly, and I'm going to talk about this later on period 6. We said the gametes have 2 forms, sperm cells and egg cells, and we use them for sexual reproduction, and other animals use them for sexual reproduction as well. Some people reproduce by just making identical clones of each other. That does happen. It's called asexual reproduction, but we can talk about that briefly later on. Let's instead talk about, let's go back. Last lesson we talked about transcription and translation, which is essentially how we go from this DNA molecule to a complex organism. We talked about how RNA can swoop in and copy half of the DNA strand, and then that little strand of RNA, that messenger RNA, it's sent out of the nucleus of the cell and goes into the ribosome. What does the ribosome do with this RNA? Well, it takes this messenger RNA and it reads the message. And as it reads the message, it strings together amino acids to make a protein. Now, there are certain codes that come together, and those codes can lead us to figuring out what type of amino acids are going to be put on. So for example, we can look at UUU at the top. That's going to make the amino acid PHE, or we can look up what that is on this page here. And last but not least, I said that some cells, obviously cells don't read the whole genetic code, they only read the parts that they're interested in. Hair cells are only going to read the parts of the code that talk about hair. Skin cells are going to read the parts of the code that talk about skin. Blood cells are going to read the parts of the DNA about blood. Then, because ultimately we switch off genes we're not interested in. Now, mitosis, what is it? Why do we need it? What's going on here? Mitosis is about making new cells. So why do we need to make new cells? There are 2 main reasons why we need to make new cells. The first is repair. Over time our cells die or they get damaged. This can be caused by cuts or sunburns, scrapes, bruises, but it can also be due by, cells getting overused. It can be caused by muscles getting, tearing cells. It could get caused by a disease or something that kills a bunch of cells in your body. But also, as I said, overused. If you, obviously a blood cell can only work for so long and then eventually it's going to die. Now, the other reason why we need new cells is for growth. If we want to make someone taller, you need to make more cells in your bones, your skin, your muscles, so new cells are needed. Same thing for if you're trying to grow a plant. you're trying to grow a plant, it's going to have to make new cells, like new leaf cells, new stem cells, well, new cells in the branch, so on and so forth. So why do we need these new cells? Mostly to replace old cells or to create new cells to make a system work. So mitosis is how the body makes new cells. So that's just the start. Okay. If anyone says, why do we need mitosis? It's to make new cells. Okay. How does mitosis work? Okay. I'm missing a pencil here. But I'm going to just ignore that. To just.
Sir, I haven't finished the last slide.
Okay. Hold on a sec. I'm not sure if you can see me doing this, but yeah. Well, if you haven't finished that, I'll keep this up for a little bit longer, but remember, you can rewatch this later on and pause it. But ultimately, I just want you to be able to take away that we mostly use it for two reasons. Repair and growth. I believe it's something like your body, I think it's like about 6 million cells die every day in your body. And that's, you know, a huge number of cells dying in a day, but that's, you know, that's pretty much how it works. I'm going to post this at the end of the lesson, so don't stress if you miss out on a little bit. I am going to move on now. So Alethea, if you've missed some of this, you can go back to it later on when I post it online. So don't worry too much. I'm going to jump ahead now to this. Why, where is it? Mitosis is the act of splitting a cell into two new cells that are exact copies or clones of each other. Okay. Now, before you can copy the cells, before the copy can happen, the DNA is first bunched up into packets, chromosomes. We keep saying this. Remember, this is why we use that picture of a box. The reason we use the picture of a box is because we wanted to remember is before we ship out the chromosomes, we want to wrap them up into a small little container. The chromosomes are copied. Now, what happens is when the chromosomes copy, we're going to talk about that, how DNA copies in a couple of seconds. But you can see on the right here, this is the process happening. We're starting off, luckily for you guys as well, I only found this out a couple of hours ago. You guys don't need to know the technical terms of this, how this spreads out. If you were in maybe the advanced class, you would, but you guys just need to know that it splits. We take this, we can see the DNA here. Can you see how the DNA is all like, you know, like a bowl of spaghetti. It's just sort of all random at this point. Then what we end up with is we end up with chromosomes. So now we've copied them, we're making chromosomes great. Then these chromosomes start to split. They all line up in the middle and then they start to split from either side. As these split, one goes left, one goes right, and then you end up with two cells. And you can see here, they're about to sort of uncoil and turn into spaghetti in there again. Why we care about mitosis is because ultimately every cell comes from mitosis. Just a quick point, I want to talk to you guys about this. This is actually not on the slide, but I guess while people are copying some of this down, one cell can turn into two cells, right? And that two cells, so one cell can turn into two cells. And each of those cells can turn into two cells. And each of those two cells can turn into two cells. So we actually end up going from 1 to 2 to 4 to 8. And you can quickly see why this is a really good way to make DNA. because ultimately what happens is that as time goes forward, the amount of cells that you have increases at an exponential growth rate. It just gets more and more and more and more and more. And this is how you're able to start, like a baby is able to start off with one, no, you don't need to copy the diagram. This is how a baby can start off with one cell. Like a baby is just an egg and a sperm cell meeting. But then that cell copies itself. It divides in half. It divides in half again. And before you know it, after a couple of seconds, you've already got probably 8, 16 cells. And, that's how you end up being like a trillion cell organism that we are now. Now, this process here of splitting is called cytokinesis. And this happens when each new cell gets one copy of the chromosomes. And essentially, it's when the cell splits. Now, that is the coolest word I have heard in a long time. I love hearing new words. Cytokinesis sounds really cool. And the fact that my body is doing cytokinesis right now makes me sound like some kind of badass, even though it's not really that. It's pretty nerdy that I know that fact. I'm going off tangent, sorry. But yeah, this is the process of mitosis. So let's talk a little bit about the last 2 facts before we finish up the lesson. There's actually not too much in this lesson. We talked about why mitosis is important for repairing and growth. We talked about how it does it. You take one cell, you copy the DNA, you split the cell, you get 2 copies of the cell with all the DNA. So we're going to talk briefly about this. How do you copy DNA? Well, the advantage is to us. We actually don't need to do much for this. We've already done this. We've already discussed copying DNA, so this shouldn't be any different. Step 1, take a DNA, let's take a DNA molecule. Beautiful, excellent. Step 2, we want to unzip the DNA, just like what we did with RNA. Step 1. Step 2 is we find matching base pairs. So the matching base pairs move in and copy the one side of the DNA, but on the other side of the DNA, we're also going to do a copy. We're not just going to go copy one side, we're also going to do a copy of the other side. And essentially, we end up with this. We will end up with 2 copies of the DNA strand. Why do we end up with 2 copies of the DNA strand? Because we're copying each strand. We're copying the left side and we're copying the right side. That's the important part here. Now, and then those copies are identical. We know those copies are identical because we know that T will always bond with A. We know that C will always bond with G, so we can easily copy the DNA. And that's actually why DNA is does such a good job of creating these copies. Why it's such a good way to move information is because we know that it's so easy to copy, because essentially only certain parts will go with certain parts. Now, what I would like to maybe quickly draw down the bottom here is your own little quick explanation of this. How I would do it is this. Let's do 1. Let's start off with a DNA strand joined together. Then we move to 1, the DNA unzips. Then we're going to move to 2, which is the matching base pairs move in. And then I'm going to draw maybe like this, and maybe like this. And then I'll do the same thing on the other side. It doesn't have to be too precise. I'm going to draw another line like this, and I'm going to do this and this. And then the last step is that we end up with 2 strands of DNA. I'll give you guys a second to copy this down. While you're doing that though, I will just quickly talk about some other random bits and pieces. You'll notice
Wait,
that
sir,
yeah,
can you explain the after the unzip?
after which bit?
Like after the after the no, after the DNA unzips.
So well, after the DNA unzips, we then have a bunch of base pairs move in, and those base pairs move in to start to copy the other side of the DNA. They start to move in, and they start joining together. And as they join together, they then eventually lead to a whole brand new copy of the DNA. All of these,
Okay.
all of these here, these have moved in and clicked into place. It's pretty much like Lego. Oh, this bit only fits with this bit, it snaps into place. Now, you'll notice that we're not dealing with uracil anymore. That's because we're making a copy of DNA, not RNA. So therefore, we're using T, not U. So that's something important, just to make sure you don't forget that in RNA, we use uracil instead of thymine. So that's how we copy the DNA. I'm going to move on because I want to keep this quick, because ultimately it's a pretty quick lesson. So the last step is, and I'm going to warn you, this is the part that's a bit weird. This is the chromosome part. Now you don't have to draw this, because believe me, even just creating this on PowerPoint took me a while. And it's also a bit confusing because the textbook doesn't sort of say the same thing that every single website sort of says about this. So it's a bit annoying. So I'm going to try to explain this. First things first, we know there are 46 chromosomes just before copying. They only have one strand of DNA, because that's what DNA is. So what we can actually refer to is they have one chromatid. This is a new word. I'm going to explain what this means in a second, but there's only one chromatid. Now each chromosome has a pair, and we're going to use a word that we've used before, which they are homologous. And we can also refer to these as diploids. I'm going to explore what this means in a second. But let's just start off with this. There are 46, okay, before copying. We count all these, 1, 2, 3, 4, 5, 6, 7. There should be 46, unless I've made a mistake here, but there should be 46. Each of these has a pair. These two are a pair, these two are a pair, these two are a pair, so on and so forth, all the way down into maybe these two here. Now these two here would probably be the sex chromosomes. So we'll have an X chromosome here, and maybe this is a Y chromosome here, or something like that. Okay, oh well, actually, because it's the same size, we'll probably say that's another X chromosome. So this is obviously a female person. Now, each of these pairs are called diploids. We call these a diploid because there are two in the pair. Di meaning two. Two in a pair. You could have something like a tetraploid, which would have 4 in the pair, or a haploid, which only has 1 in the pair. But at the moment, for us, we've got these are in pairs of 2. Now, I don't have a slide for this because I ran out of time and I realized that I was running late, so I thought I'd better just teach this as it is. But just before, right, I might write this down as the thing. Each before splitting, each chromatid is copied. So when each chromatid is copied, each one of these halves makes a copy of itself. And I'm actually going to do this for all of these, just so that we can get an idea. This is going to take me a little while, but this is starting to look a little bit more like, whoa, the picture that we saw for a karyotype. Okay, each chromatid is copied. Now, this part is the weird part. We would say that there are 46 chromosomes. But now each of those chromosomes are made up of, each chromosome has 2 chromatids. So what that will mean is that for the cell, like one copy will go to we'll just call this the left cell, and one copy will go to the right cell. And that will happen for all of these. And the reason why they'll happen for all of these is then we'll have 46 going one way, 46 chromatids going one way, 46 chromatids going the other way. Question for you: how many chromatids are there in total at this point right now? Can someone tell me how many chromatids are there exactly at this point now?
92.
92. At this point here, there are 92. So before splitting, each chromatid is copied. There are 92 chromatids, making up 46 chromosomes. And each chromosome is in a pair. So there are 23 pairs. That's why these make these weird X shapes. By the way, there is a technical term here. This part in the middle where they join, it's called a centromere. But you don't need to know that. So I'm just going to get rid of that because I don't want you to get more confused than you have to be. So, but yeah, it's called a centromere. And that's how we use to count them. The reason I can count there are 46 is because there are 46 X's. There are 46 crosses. And I can tell that, and each cross is made up of 2 sides. Okay? That's it. That's all I had to explain about, because once those things split, going back here, once those things, make copies of themselves, they all line up in the middle, and then they split. One half of the chromatids go to the left, one half goes to the right, which means that you end up with 46 chromatids here, or 46 chromatids here. Or, because they're on their own now, we would refer to them as 46 chromosomes. It doesn't matter. So, ladies and gents, what I want you to do, first things first, I would like you to, in chat, I would like you to give this lesson between a 1 and a 5. Do you think you understand this so well you could explain this to someone else, maybe someone who missed the class? Do you think you understand it pretty well, but probably wouldn't be able to explain it? Do you reckon you might need some help? That's a 3. Do you reckon that, you're probably going to need a fair bit of help? I'll probably have to send you an email. That's a 2. A 1 means you have no idea what's going on here, really. You're sort of like, I know the words, but I don't understand how this all comes together. And a 0 means you pretty much just have no idea. You've been asleep the whole time. You've just woken up. All right. I'm seeing a lot of 3s, which is good.
I'm on like, I don't know, like 2 or something. I'm lost.
What part do you reckon, Brandon, what part do you think confused you the most?
This whole entire thing, this chromosome, the only thing I found interesting was when, like, you know, that big word thingy. You said that word was like the coolest to you.
I don't know.
I don't
just
even
like
understand what that word is. I don't understand all of this stuff.
Okay, well, like, okay, yeah. Well, you might need
I
to.
understand the words, but like I just don't understand what you're on about.
You don't understand. Okay, why? Here's the question for you. Why does a cell? Why did DNA get coiled up into chromosomes in the first place?
Wait, what'd you say?
Why does DNA get coiled up into chromosomes in the first place?
You see, I don't even know what chromosomes mean.
Okay, so a chromosome. This is the first part. If you miss this part, even if I just get a little bit to you, the chromosome. You know, with DNA, how it's a big long string.
Yeah.
Right? It's a big long string. If you were trying to move a big long string from one place to another place, it'd be really difficult. It's like, imagine trying to drag a rope around the neighborhood, you know, just dragging it along the ground. It gets stuck on things, people would trip over it. So what you would do is you take that rope and you'd coil it up. So instead of having a big long rope everywhere, you probably coil it up into a nice little thing, and then you could walk along with that without anyone tripping over it. Does that make sense? Yeah, that's exactly what DNA does. Instead of DNA being this big long string going around in the cell and tripping everyone up, it coils itself up. And when it coils itself up, we call that a chromosome.
Oh.
So that's all that is. All a chromosome is just a big old tied up piece of DNA. It's like imagine you're taking that big long rope and you wrapped it up into a nice little thing that you can carry. That's what a chromosome is.
So 46 chromosome is like 46 ropes tied up.
Exactly. So you've got.
Okay, cool.
So imagine now, instead of having one rope, you've got 46 of these ropes, 46 of these strings. So now you definitely need to pile them up. You've got, you'll end up with 46 little bundles of rope. And that's why, humans happen to have 46 bundles of these ropes. Now I'm going to take this analogy to a little bit further, right?
Yeah.
Some of these ropes are really long, okay? Some of these ropes are really long. Some of these ropes are really short. But what's interesting about DNA, right, and about these ropes is that they come in pairs. The pairs are always the same length. So, and you can tell that these strings are the same length because when you coil them up, the long ones coil up into big coils, whereas the little ones are only coiled up into little ones. Imagine trying to coil a little bit of string around your hand. It's going to be able to put it in your pocket. If you're trying to coil up a big long piece of rope, then it's going to get, it's going to get in, you won't be able to fit it into your pocket. So, and yeah, that's the next main idea, is that chromosomes come in pairs. There are 23 pairs. So for every piece of rope, there is another piece of rope that's the same length. The final part, I guess, the part that we were talking about today is that when you want to just before you end up to copying something, you take each of these ropes and you get another piece of rope and you match it to be the same length as the first piece of rope. Yeah, are you with me so far? Have I lost you?
I'm with you.
So now you've got a bunch of these green rope with every one of these blue rope. You've got so now
Wait,
I've got.
is class over?
Yeah, if you want to leave now, I'm just sort of explaining a little bit extra. And then all you do to make a new cell is you say, right, all the blue rope, that's going to go in this box. All the green rope, that goes in this box. And because you've made sure that each of the ropes is the same length, you can be sure that you've got exact copies. And then, yeah, that's how you end up with two cells. And that's pretty much just what I talked about all through today's lesson. So hopefully that made it maybe a little bit more clear. At least now you know what a chromosome is. It's just a big pile of DNA that's been squashed up, like wrapped up like a rope. Anyway, ladies and gents, what I need you guys to do is your homework is to the rest of this lesson, and I'm going to talk. I haven't decided, but I'll probably meet up with you guys this afternoon for a little bit of a lesson, maybe not a full lesson. I'll let you know then. But I want you guys to work on 1.2. All right. Sorry, 2.1 in the style. I think I've made it open and available to everyone. So that's something I want you to do. It's very, very short because I got rid of a lot of the complicated stuff at the end. It's pretty much what we just talked about today. You got any questions? Let me know. Otherwise,
Wait,